A quick disclaimer before we begin this Chapter 3 video. This chapter is very organic chemistry and biochemistry focused. Keep in mind that these are not my primary areas of study, and I will only be going as in-depth as the book goes for brevity and clarity purposes. So as we continue, please keep that in mind. Hello and welcome back to Biology for Beginners. As always, I'm your host, Morgan. This week, we're going to be talking about proteins. As always, we're going to pause here for a moment before we dive into the meat of the video so that you can grab the terms and definitions and be fully acquainted with the vocabulary we will be using today. A quick reminder, you can also grab this and the rest of my PowerPoint presentation in PDF form over on my Patreon page for absolutely free. That link is in the description below. What exactly is a protein? Now, I'm sure that when you Google protein, you're very used to seeing images like the one I have on the right here showing all sorts of proteins that you can eat. But what you probably don't see is this very confusing image to the left that is a computer-generated representation of what a protein actually looks like. And proteins are molecules made up of things known as amino acids, and these keep your body functioning. Proteins are the things that make up enzymes that help the chemical reactions in your body take place so that you can function and be you. Proteins are also used for transporting things across cell membranes. They're useful in signaling and recepting hormones and other signals being sent from your brain and other parts of the body. They're important in defense, especially in the immune system. They help with structure and movement. So proteins are the things that pretty much help to make life happen. Pretty much, if there is a function in your body, there's probably a protein helping to make it happen. So if proteins have so many different functions, what exactly makes them function that way? Well, it's their specific shape. The shape of the protein is going to determine its function. So we'll get a little bit more into how they get that shape, but a protein basically folds into a particular shape, and because of the way it's shaped, you have the active sites and the places where these reactions, the reception of different signals, all those things are going to take place. And if the shape is incorrect, the protein can then not function. That shape is how enzymes will recognize the materials for the reactions that they catalyze. So shape determines function, especially in proteins. This is important because when they denature, that shape is then lost, and therefore that function is lost. And any incorrect folding along the way will lead to an incorrect shape, and thus an incorrect function or no function at all. And you'll just have a protein floating around that can't really do anything. The things that will cause it to denature and break down are changes in the pH, changes in the salt content, if the salt content in the surrounding area is not appropriate, and high temperature, which is kind of why you run a fever when you're sick, because your body is trying to denature any proteins that they have determined are not part of you. But that is a massive oversimplification of that amino response, and that is where I'm going to end that one for today. But shape determines function, and if something is thrown off, the shape will change because the protein will unfold and break down, and the function will no longer happen. So what exactly are amino acids, especially if they're so important to this whole protein shape thing? Well, amino acids are the little monomers or the building blocks of proteins. They have these R groups that have many different properties. The R group, it really just stands for what the heck is attached to them. Amino acids are made up of an amino group and a carboxyl group, with the R group hanging off that carbon in the middle shown there. And when you join two amino acids together by a dehydration reaction, you get what is known as a peptide bond, and you form polypeptides. However, if there's only two amino acids joined, you get a dipeptide, di-2. Any more than two, you will get that polypeptide. However, it's still going to be a dehydration reaction. It's still going to be another peptide bond formed. And you can see that depicted down below. There are 20 amino acids that make up the proteins in your body, and I have a lovely little chart here on the left for you to look at if you are interested, showing all of those R groups that can be attached to them and the naming that goes into the amino acids. I am not going to get very in-depth on that here, since the book does not go that in-depth. It just kind of mentions that they're a thing. I just think it's useful to show them as well. And I had to look at them. I had to memorize them. You guys at least have to look at them. Moving on here. The specific order that these amino acids show up in that polypeptide chain matters. They're kind of like letters in that case, where I can have the letters D-O-G, and in that order they spell dog, but if I rearrange them, you get G-O-D, which is a very different word and means a very different thing. Amino acids are similar. In a specific order, they will end up making a specific protein, but if you rearrange them, they will make a different protein. And these R groups are what are going to determine that protein shape. And this is done through the way that their hydrogen bonding work. The more hydrophilic protein or amino acids, when the protein folds together, will end up closer to the outside, with the hydrophobic ones on the inside, and other reactions that they have between each other as well. But we will get more into that folding in the next few slides. So protein structure can be broken down to different levels. And we're going to start with the primary structure. And this is the specific order that the amino acids are in the polypeptide chain. So from the first to the last in a straight line, that is the primary structure. Just what order those amino acids would be in if you unfolded that protein and held it in a line. The next level of structure is going to be the secondary structure of that protein. And this is going to be that specific folding or twisting of the chain in these little local areas. And when you get this, you either get the alpha helix on the left or the beta pleated sheet on the right. And these are going to be held together with hydrogen bonds, which are what is depicted by those little dotted lines there. And you can have alpha helixes and beta pleated sheets in the same protein. These are just the structure at localized areas, not necessarily the whole protein. So the last two parts of structure are going to be tertiary structure and quaternary structure. And I've lumped them together in one slide because they are fairly similar. And the tertiary structure is just the shape due to the R group interactions. So once you get those secondary structures, the R groups are still going to interact with each other, and it's going to fold even more, putting those helices or sheets in different places. That is your tertiary structure. Quaternary structure then takes two or more polypeptides and gets them interacting, making up the greater protein. However, it is important to note that not all proteins have quaternary structure. But here we have pictured is going to be hemoglobin, the polypeptide, the protein that is important in your blood. And it is just a good depiction of that quaternary structure there. So just a quick recap here on the protein structure. Primary structure is just your straight chain of amino acids and the particular order they're in. Secondary structure is the local folding and or twisting of that chain, giving you the alpha helix or the beta pleated sheet. Tertiary structure is then how those R groups continue to interact and fold those secondary structures around. And then the quaternary structure is two or more polypeptides then interacting to form that larger protein, and not every protein has quaternary structure. Well, that's all I really have for you on proteins today. I will now stop the biochemistry torture I have put you through for this week. You will be happy to know there's only going to be one more chapter 3 biochem video next week, and we will move on from this hell that I have put you through. But I did have to go through a whole quarter of going much more in depth than we have gone in these videos, and proteins are weird, especially when you go beyond what I have just told you. But I will now release you. I hope you have a wonderful rest of your morning, day, evening, what have you, and I will see you all next time. Quick reminder before I go, if you want the PDF version of my slideshow, please pop on over to my Patreon that will be linked down below, and the PDF is absolutely free. Thank you again for watching this video. Don't forget to like, comment, subscribe, and ring that bell if you would like to see more. And if you'd like to follow me on any of my other social medias, the links are down in the description below. Don't forget to check out thereptilegoth.com for all of my articles and blog posts. If you found any value in this video and you would like to help support the channel, please check out my Patreon page. That link is also in the description down below. And a special thanks goes out to my Diamond Dragon patron, Diane V. What you're doing is really helping me fund a dream here. I will see you guys all in the next one.